international research CRIB South Korea in 2011 for his contribution to the field of plant taxonomy. In nutshell, I want to tell you he is a young, energetic taxonomist of the country, and probably many young students he may train in molecular systematics, similarly in classical taxonomy, and uh, he will make good progress in, growth for taxonomy in India. On behalf of IIT and our Dr. Shashidharan, who is our president, I request Professor Nampi to confer the award and certificate to him through presentation on a screen. Right, so this is a certificate. This is a gold medal, Sivarajan gold medal, which is a very prestigious medal for any taxonomist. Now I request Dr. Ritesh Chaudhary to deliver his talk.
Thank you so much, sir. I hope I'm audible there. I would like to.
Yes, you are audible, very much audible.
Thank you so much, sir. I would like to upload my PPT first. It's getting uploaded. Thank you so much, sir, for the good words. Still getting uploaded. Almost done. Just a minute.
Yeah. So you know you have been allotted 30 minutes by the organizer. So take care of time also.
Sure, sir. Sure. So before I start my presentation of the talk, I would like to dedicate this award to all my gurus with a shloka. Prereka suchakshveva vachiko darkashtatha shikshako bodhakascheva shadeti guruva smita. The inspirer, the informant, the guide, the educator, and the seeker, all of them are the gurus. And I have divided my talk into two parts. First is my tryst with plant taxonomy, my research, and the challenges which I faced during all this period, and the way forward to it. So my research journey, as the others are already informed, that my research journey actually starts from Botanical Survey of India, the then Arunachal Field Station. Now it is known as APRC, Arunachal Pradesh Regional Center. So after finishing my MSc from Bhagalpur University in Bihar, which is my hometown also, I joined BSI as a JRF, and I was assigned the work Flora of Upper Siang district of Arunachal Pradesh. You can see the district is highlighted there, bordering China, and it is important for two reasons. First, it is a micro center of endemism, and for its ethnobotanical wisdom. And the area is quite huge, 6,118 square kilometers. And no floristic details studies were available except a checklist by Dr. Rao and Joseph in 1965, where he had reported 319 species of angiosperms from the area. The area is also very important because two of the very important protected areas lies in the district called Dihang Dimang Biosphere Reserve and Mauling National Park. As far as the ethnobotanical wisdom is concerned, district is home to three major tribes called Adi, Memba, and Khambas. And the area shelters some of the very, very rare and medicinal species such as Aconitum lithale, Coptis tita, etc. So many. But the terrain is not so easy to approach. As you can see in the picture was taken in 2004 at Yinqiang. This was the entry point of the district, where you have to cross the bridge, cross the Siang River, and after that you can get a vehicle to move further. And there are several other places which are very botanically very rich but difficult to approach. In 2004, I almost spent 5 years, more than 5 years, doing this work. And it was really challenging for me because going from a plain land and working in a hill area was really very challenging. So sometimes you have to walk a lot, and you are not provided with any help. There you have to do everything alone, sometimes with the forest officials, sometimes with the porters provided by the government. And if you're lucky enough, you will get some conveyance like this. So that way it was very challenging, but still we could document more than 1,000 angiosperms from the area, documenting some very rare plants like Paphiopedilum venustum, Studnera assamica, and so many orchids, almost 88 species of orchids we have reported, and lot many plants of ethnobotanical importance. And lot many interesting Himalayan endemics like Aconitum lithale, which is used in the arrows of the tribal people, the local communities, Coptis tita, which is a panacea for all sort of diseases, and this unique cucurbit you can say called Indophavelia khasiana. This is probably the only image available on net, which was taken by me at India-China border, called Galing. So during this period, we could document 1,003 flowering plants, one new species called Lembelia arunachalensis, 3 new records to Indian flora, 12 new records for Arunachal Pradesh, 1 recollection, 73 endemic plants, and many first-hand information on the ethnobotanical uses. And this is the paper which we have published, a checklist of the district. You can download this freely available on my ResearchGate profile also. Also, we could document the ethnobotanical wisdom of the area in a paper which is also available on my ResearchGate profile. Other than that, we could publish so many other papers while working there, along with my PhD guide, Dr. A.K. Das and Dr. R.C. Srivastava. While doing this, in 2007, in Department of Botany, Bhagalpur University, there was an international workshop on molecular systematics, which was organized by Dr. A.K. Pandesar. And he had invited a wonderful lady, very dynamic lady from University of Vienna, Austria, Dr. Rosabelle Samuel. And she, on this workshop, actually trained me, this was the workshop where I learned the ABC of the molecular systematics. And this workshop probably changed my thinking completely of how biodiversity research should be done. I found that just documenting species is not enough. There's several shortfalls. When we are conducting a biodiversity study, there are several shortfalls called Wallachian, Prestonian, Darwinian, Roncarian, and these several shortfalls. Like if you don't document all the species, then there is a shortfall called Linnean shortfall. If you don't document the geographical distribution, if you don't document the population, if you don't know the evolution of this species, there is a shortfall. So this completely changed my thinking about plant taxonomy. And I was fortunate enough, once I finished my PhD, I got a postdoctoral position in GKVK Bangalore on a project which was handled by Dr. M. Sanjapa sir, the then BSI director. And I had to shift to Dr. Uma Shankar's lab, which was a huge lab, so many people working there. And there I was involved in some 3-4 projects as a plant taxonomist. And one of the projects you could see that first thing, it was like identification. A major project which I was involved in was identification of high yielding and alternate source of a few important metabolites from herbarium material. And also a project on DNA barcoding and secondary metabolite screening of Calamus species in India. So for collection of these Calamus species in India, along with my colleague, Dr. Senthil Kumar, who is presently associate professor there at SRM University, along with him, we could travel to Andaman and Nicobar, starting from Diglipur, the northern point of Andaman, to the Indira point in Nicobar. Almost 40 days we were there. We could collect all sort of Calamus growing there and all endemic species, and also we made effort to barcode DNA barcode all those Calamus species. Other than that, another result came out along with Senthil, Dr. Senthil, who is a friend and colleague at that time. And we could produce a work on the role of ratans, a role of Calamus species among Mongoloid tribes and settlers of Andaman and Nicobar Islands. This paper is again free to download. You can see and download this paper. You can go through and you can find like how important ratans are there for those people, the local people. Just within a year, I got a postdoctoral fellowship by NRF, National Research Foundation of Korea, in 2010 itself. And I got the opportunity to join Korea Research Institute of Biosciences and Biotechnology, where I still refine, where I learn the molecular systematic tools and techniques under the guidance of Dr. Junku Lee. There, I was involved in two major projects. First project was related to molecular phylogeny of Polygonum sensulato, and another project I'm going to discuss later. The first project on molecular phylogeny of Polygonum, and as you know, Polygonum is very complicated genus at that time, and even Hooker in Flora of British India, he has said, it's a very troublesome genus, and I cannot hope that I have finally settled the limits of those, especially of the Persicaria and Avicularia sections. We use molecular tools to resolve all these problems. And first time we could resolve taxonomy of the genus Conigia. We could also treated Persicaria in two major taxonomic categories with and without capitate inflorescence. And common lineage to the Persicaria and Ablygon section was also proved, and close linkage to Phagopyrum genus was also proved. And we could provide high resolution phylogeny to Polygonum sensulato. And the results were published in Genetics and Molecular Research Journal. The other project in which I was involved was Flora of Honban Nature Reserve. You know, this nature reserve was in Vietnam, and Vietnam till half of the 20th century, because of decades of war with USA and other countries, this was in complete isolation. And the country was completely closed for foreigners. I was fortunate enough to join this Korean team, and they had just opened Vietnam for floristic research. So I got the opportunity to work on an area which falls in Mekong River watershed, where in the last 10 years, more than 10,000 new species have been discovered. Not only this is the second richest biodiversity hotspot in the world after Amazon. And this stretch has more species per unit area than any other river in the world.